I'm so very excited to present to you today Dr. Neil Theise. Dr. Theise is a diagnostic liver pathologist and adult stem cell research at Albert Einstein's Beth Israel Medical Center, where he is a professor of pathology and medicine. Through his research, he has contributed greatly to our understanding of liver microanatomy and stem cell regeneration. Dr. Theise is credited with the discovery of the biomechanistic pathway through which bone marrow stem cells differentiate into liver cells. He's considered a pioneer of multi-organ adult stem cell plasticity and has widely published on the topic in Science, Nature, and Cell. While continuing his lab research into liver stem cell regeneration, Dr. Theise has applied his empirical reasoning to investigate theoretical biology and complexity theory, suggesting alternate models of the body other than cell doctrine. He has most recently edited and published Longevity, Regeneration, and Optimal Health, Integrating Eastern and Western Perspectives, a collection of 31 essays by researchers such as recent Nobel laureate Elizabeth Blackwell and His Holiness the Dalai Lama. Today, Dr. Theise is kind enough to come to Touro to speak with us about his research-based investigations into how meditative yogic and related practices can assist in stem cell regeneration. So let's give a warm welcome for Dr. Neil Theise.
Thank you. Thank you for inviting me, Rachel. I have like 5 hours worth of material. Don't worry, I'm not going to give it all to you. So I was trying to decide which bits to focus on, and I could do the details of the physiology of how yoga and meditation hit your stem cells, but maybe you'll have me back if I don't do a horrible job. Because I sort of want to instead call into question everything you're learning in osteopathy school, and everything the physicians you're talking to are telling you is probably only partial view. And so this is what I refer to as my hand grenade talk, because it's just sort of like throw it into the room, and you'll respond as you will. The most important thing I'm going to say, probably I'm just going to say right up front, and it's actually a quote from Goethe from his Maxims, where he said, "Thinking is better than knowing, but looking is even better." And as Cheryl is teaching you, where's Cheryl? She's somewhere in here. Oh, there. Oh, the Dr. Schiller seat. No, he'd be sitting in the back. Yeah. From a pathologist standpoint, this is our practice. Even if you give us the clinical history beforehand, we have to look at the slide and really look at the slide to see what's there, because clinicians often have no idea what's there. And in liver disease, that's particularly true. And in fact, habitually, if I get a liver biopsy, I don't look at the clinical history before I assess the slide, because any bit of clinical information is going to bias how I'm going to interpret the very subtle things that happen in the liver. It only has a very limited range of how it can respond. So it's easy with any given clinical history to see that disease that they're expecting in the biopsy and make a case for it, even though it's something else altogether. So this is sort of standard practice of pathology, and it should be standard practice for all of you when you go into a room with a patient. You may have all sorts of preconceived notions because of what you've told, what you've been told about the patient, or what the patient tells you, or just what you think when you walk in a room and see a patient there and you make judgments about them and assessments before you even begin your exam. It's about having, in what we call in Zen practice, beginner's mind. Come in, drop all those preconceived notions. You may need to bring them back in later, but look at what's in front of you. That's the key thing. And so what I'm going to tell you is sort of the story of what's happened to me over the last decade, because when I trained with Cheryl at Mount Sinai, the last thing I ever thought I was going to be talking about was stem cells complexity and Buddhist metaphysics and stuff like that. I was just a liver pathologist. Oh, sorry, I've input some slides and the color shifted around. This is a paper from a mentor of mine who used to be at Mount Sinai, who trained the senior liver pathologist at Sinai and then became dean of Tulane Medical School, named Michael Gerber. And in 1996, which you can't see, he died shortly after this. And so this was one of his last papers in liver pathology. And he was one of the few people who believed livers had stem cells. That used to be a very controversial notion. And what he pointed at in this paper, this is normal human liver. And if you stain for biliary type cytokeratins like keratin 19, you get bile ducts staining positively for keratin 19. But then you have these small little cells out here amongst the hepatocytes. And he suggested that these were stem cells for human livers for a variety of reasons. The problem is, and seeing this paper, the question that arose in my brain is, this cell is in a perfect place to regenerate hepatocytes, but stem cells for human livers, for any liver, theoretically also have to regenerate the bile duct. So how is it going to get from here to here? It's got to either travel and digest through stroma, which makes it sound like cancer, which we know it's not, or there's got to be something else. And the short version of this story is because I just was irritated by that question of how that cell is going to get there, I started doing serial sections on normal livers and staining them for keratin 19. And so we see here a normal human liver, portal tract with bile ducts and ductules, and there's one of these cells over here. And the next step section down, 4 microns down into the tissue, ducts and ductules, and there's that cell. Next level down, there's that cell again. Next level down, there's that cell. Here's a bunch more coming in. But these cells are about 6 microns across. So either it's a long spindle-shaped cell, which makes no sense also for a stem cell in the liver, or it's a chain of cells that are actually lining up. And sure enough, here they are again at the next level down. And you see them come together and link up and become a bile duct. And so this structure had not been described in any of our histology textbooks. And when I first saw this, I actually took it to Swan and said, Swan, look at this. There's this new structure here. And she goes, no, there's not. And I said, look. And we went through these serial sections and goes, well, I don't know what that is. I said, okay. It turns out there's something called the Canal of Herring. Did you learn about canals of Herring in histology? Oh, good. Anyone want to tell me what a Canal of Herring is? This is an exam. It's the link between the hepatocyte canalicular system and the bile ducts. It's how bile gets from the hepatocytes to the bile ducts. And the way you may even have described it is that it sits at the limiting plate. Where's my arrow? There we go. If this is the edge of the stroma of the portal tract, the classic view that Herring described and drew pictures of in 1865, and no one ever revised until I came along with this stupid stuff, said that it comes in, the canaliculi drain at the margin of the portal tract. But it turns out that these thin little projections that with these small cells lining up are the Canal of Herring. And the Canal of Herring actually extends out into the parenchyma like this. Big deal, right? Who cares about bile ducts? Well, so if this is how we would diagram a normal liver, this is now how we now have to diagram a normal liver, because the canals of Herring are branching almost like wire brush structures into the parenchyma about 1/3 of the way towards the central vein. And why that becomes important is that in fulminant hepatic failure, let's say from a Tylenol overdose or massive necrosis from hepatitis A or hepatitis B, when the inflammation and destruction gets widespread enough, spreading out from the central vein and widening circles to hit the Canal of Herring, that acts like a tripwire, and it turns out the Canal of Herring is where the new liver cells start to grow from. So with this study, we showed that the liver not only had stem cells, but Michael Gerber was right, the cell he was pointing at is the stem cell, and that in fact it's a pre-existing structure we knew about, we just didn't know what it looked like, and that's the Canal of Herring. That's the stem cell niche for livers. So that's how, I started as a pathologist worried about bile ducts, and that was like as far as my ambitions were taking me. I presented this to my chairman. At this point, I was at NYU, and I said, I think we know that there are stem cells in the liver. I think we've actually proved it, and this publication of this paper is considered the proof that human livers have stem cells. So I said, I think we know where they are, and I think we could isolate them. Any scientists in the department who would want to research that? And he said, well, it's your idea, you should research it. And I said, I don't want to research it, I'm a diagnostic surgical pathologist, I don't want to go into a lab. And I laid down conditions to apply for a grant that I thought would be unmeetable. I said, well, if I get money for a grant to go on a sabbatical to study this, you have to promise to hire someone to do all my clinical work while I'm away so my colleagues don't hate me when I come back. And he actually said, okay. So I reluctantly applied for a grant, figuring as a pathologist I wouldn't get one. Unfortunately, I got one. And then I went off to Yale to explore the Canal of Herring. And this is where the fun stuff starts. So the Canal of Herring hypothesis at that point explained all the animal data on liver stem cells except for one experiment. And without going into the details of that, I thought, well, one way to explain that one errant experiment was to suggest that some stem cells came to the liver from outside. And where would they likely come from? Well, if they're coming through the blood, maybe they come from the bone marrow. And a wrong assumption I made was, since the hematopoiesis starts in the liver, maybe it's got an affinity for bone marrow. And so that justified the question in my mind, although it turns out that's irrelevant. And so I had this idea that maybe there were stem cells coming to the liver from the bone marrow. And at that point, I was at Yale to study the canals of Herring, which I never actually got to do. I teamed up with Diane Krause, who's a hematopoietic stem cell expert at Yale, who does bone marrow transplants in mice. And we came up with this experiment. Take a female mouse, that's why it's sort of pink, lethally irradiate it to wipe out its bone marrow, repopulate the bone marrow with a male bone marrow transplant to prevent death from the radiation. And those mice do very well. They basically all survive with 95 to 100% of their bone marrow and all their circulating blood cells containing Y chromosomes because it's a male donor. And since that's in a female mouse, you can use the Y chromosome as a tracker for whether a cell derived from the bone marrow. And then we did something that no one had done before, although it turned out there were a few groups doing this simultaneously for other reasons. We did fluorescence in situ hybridization for Y chromosomes in the liver tissue. So this is normal male mouse tissue, mouse liver. That's a Y chromosome in the nucleus of an hepatocyte. And you see lots of Y chromosomes. Some nuclei don't have them because these are very thin sections. It's 3-micron thick sections to do this stain. And so some nuclei are not fully sampled, so we're missing the Y chromosome. Negative control tissue, female mouse liver, and there's no random red dots floating around. Here we've got a Y chromosome in the nucleus of an hepatocyte, so this is an hepatocyte derived from the bone marrow. And this was actually the first one I ever saw. I actually have a diary entry about that, and I don't even keep a diary. So this got published. This was the second paper that got published on liver from bone marrow. A friend of mine published the first one. The week he got accepted to Science, we got rejected from Nature because even though we had answered all their questions, the reviewers simply didn't believe it. He had the misfortune of getting accepted to Science, misfortune because he was a postdoc in the lab of the chair of pathology at the University of Pittsburgh. And while his chairman had let him do the research, he didn't believe the research and would only let him submit the paper for publication if he took his mentor's name off the paper. So imagine being your first major scientific paper and your own boss doesn't want to be associated with it. At the same time, there was a paper also published in Science that showed skeletal muscle from bone marrow, and another paper that showed brain stem cells turning into blood. And so those three papers were considered by Science their 1999 discoveries of the year. We got published 6 months later, and then people started publishing on bone marrow going to stem cell stuff, which upset everyone because organ systems aren't supposed to feed into each other. So people started arguing about this, and the major critique at this point, we had all done experiments where we transplanted 100,000 cells, 1 million cells to accomplish the bone marrow transplants. And they brought up the justified criticism, well, we're really not showing hematopoietic stem cells are turning into all these other organs because maybe there's a special stem cell for the liver and a special stem cell for the muscle that has nothing to do with hematopoiesis. They just also happen to hang out in the bone marrow.